everyone, I am Dr. Sonal Chain from Department of Anthropology, University of Delhi. In this series of forensic science lecture, today we are going to discuss about immunochemical techniques. In our previous series of lectures, we have discussed various immunochemical techniques like ring test, diffusion test, immunoelectrophoresis, and in continuation to that, we will be discussing more immunochemical techniques. These techniques are useful for when we have to analyze some blood sample which has been found from the crime scene, and we have to go for the species identification of those samples, or for other identification we use various immunochemical techniques. To start with, immunochemical techniques are based on a reaction of antigen with antibody, or more exactly on a reaction of an antigenic determinants with the binding site of the antibody. That means antigen bind to a specific site of the antibody, and an antigen will bind only at a particular site if the site is in determinant with the antigen. The antibodies which are used, they are produced by various ways which are used in these different chemical reactions. So, immunoassay is a test that uses antibody and antigen complexes as a means of generating a measurable result. Measurable result means that whether antigen combines with the antibody or not, and if antigen is showing reaction with antibody, how much antigen or antibody is present. So, immunoassay methods are qualitative as well as quantitative methods. An antigen antibody complex is also known as immunocomplex. Immunoassay techniques, whenever we say, it is combined of two words, immuno and assay, where immuno refers to an immune response that causes the body to generate antibodies, and assay refers to a test. Thus, an immunoassay is a test which utilizes immunocomplexing when antibodies and antigens are brought together. So, usually whenever we are conducting a test, we have to look for, if the antigens are present, then we have to look for which specific antibody will give a positive test with a specific antigen. Only a specific antibody will give a particular test with antigen, and if the antigen and antibodies are not specific to each other, they will not give any test, any positive test or any other visible reaction. But if antigen and antibodies, they form a complex, that means they matches with each other. Immunoassays are different from other types of laboratory tests such as colorimetric test, because they use antigen antibody complexes to generate a signal that can be measured. In contrast, most routine clinical chemistry tests utilizes chemical reactions between the reagents, a solution of chemical or other agents, and patient samples to generate a test result. But specifically in the case of immunoassay techniques, we look for the antigen antibody complex which are being made, and also we can measure that how much antigen or antibody has been utilized. Immunoassay utilizes one or more select antibodies to detect the analytes of interest. The analytes are measured, maybe those are naturally present in the body, for example thyroid hormone, and those that the body produces but are not typically present, such as cancer antigen, or those that do not naturally occur in the body, such as in abuse drug. So, all the analytes can be measured, which may be the natural secretion of the body, which may be not present in the body, but because of some disease that antigen is present in the body, and sometimes it comes because of ingestion of any abuse substance if we have taken that. So, now whenever we are talking about the antigen and antibody complex, we must know what could be an antigen and what could be an antibody. Mostly the antigens, they are proteins or large polysaccharides which are from a foreign organisms. If they come from microbes, they could be capsules, cell walls, toxins, viral capsids, flagella, etc. If they are known microbes, they could be pollen, egg white, RBC surface molecules, serum proteins and surface molecules from transplanted tissue. So, antigens could come even from microbes or they could come from non-microbes too. Lipids and nucleic acids are only antigens when combined with proteins or polysaccharides. The molecular weight of these antigens could be 10,000 or higher, whereas haptens, they are the small foreign molecules that is not antigenic. It must be coupled to a carrier molecule to be antigenic. Once antibodies are formed, they will recognize the hapten, that which hapten is there, and their molecular weight is mostly more than 10,000 or higher than that. Now, antigens, they have an epitope which is present, a small part of an antigen that interacts with the antibody. The shape of the antigen or the part of the antigen which interacts with the antibody is known as epitope. Any given antigen may have several epitopes. Each epitope is recognized by a different antibody. That is why antigen reacts with specific antibodies, because the shape, the size and the chemical nature of the epitope is very different. They always react with, the epitope reacts with that kind of antibody which is in conjugation with that particular antigen. So, the chances of having false negative reactions in immunochemical assays or immunoassay techniques is very low, because antigen antibody reactions are very specific. In some cases like colorimetric tests, etc., some reactions might have some false positive or false negative test, but antigen antibody reactions are very specific in nature. If we have to analyze the blood for any species specific reactions, whether that blood belongs to human or any other animal, then these immunochemical reactions are very good to identify. We can put human antibodies, we can put the antibodies of those animals whom we think the blood could be, and the specific reactions will give us a 100% result that these antigens or antibodies belong to this particular species. So, when antigen, the epitopes are those regions where antigen regions that interact with the antibody. Depending upon the shape of the epitope, it reacts to the specific part of the antibody. If we look the shape, it could be just for visualization purpose, it could be like triangular, it could be like circular. So, the part of the epitope which is going to get reacted with the antibody should be in such conjugation. Okay, let us take an example of a jigsaw puzzle where the piece will only fit when the shape of the two pieces are exactly similar. So, antigen epitope will only react with the antibody when their shape is in conjugation and is in similarity with the same antigen or antibody. That is why we call that antigen antibody reactions are very specific reactions. So, we spoke about antigens, now we talk about the antibodies. Proteins that recognize and bind to a particular antigen with very high specificity, those proteins are known as antibodies. Usually antibodies are made by our body's immunity system in response to the exposure to any antigen. Any virus, any microbe reaction, they may have several antigenic determinant sites to which different antibodies may bind. So, if any foreign antigen enters our body immunity reaction will try to produce antibodies in response to exposure of that particular antigen. Whenever there is a foreign antigen which enters our body immune reaction, it starts working and it produces antibodies in return to that antigen. Each antibody has at least 2 identical sites that bind antigen. Those 2 sites are known as antigen binding sites, and only specific shape antigens can be binded to that specific site of the antibody. The number of antigen binding sites is the valence of an antibody. Most of the antibodies are bivalent in nature. That means they will have 2 antigen binding sites. 2 antigen can bind, just we can imagine like a Y shape, where one antigen can bind on the one hand of a Y and other antigen can bind on the other hand of the Y. So, mostly antibodies are bivalent in nature. They belong to a group of serum proteins which is called the immunoglobulins. Antibody structure, if we see, it is a monomer. It is a flexible Y-shaped molecule which has 4 protein chains, 2 identical light chains and 2 identical heavy chains. They also consist of variable regions. 2 sections at the end of Y's arm. They contain the antigen binding site, identical on the same antibody, but it varies from one antibody to another. So, an antibody structure is a Y-shaped molecule having light chain as well as heavy chains. It has variable region also, it has constant region and it has FC region. The constant region is the stem of monomer and lower parts of Y arms. FC region is stem of monomer only. It is important because they can bind to complement or cells. So, antibody structure, if we look at it moreover it looks like a Y-shaped structure, where you can see that on the left hand side it has one heavy chain and one light chain, and on the right hand side it has one heavy chain and one light chain, and it has variable region as well as constant region. We can see the at the endings of the light chain as well as heavy chains, we have antigen binding sites, and the shape of these sites determine that which antigen will come and will bind at these antibody sites. So, the antibodies, they are also known as immunoglobulins. Immunoglobulins are of various types. They are found in our body and they have different functions. So, let us talk about the first immunoglobulin, which is IgG. Its structure is monomer. It has 80% percentage of serum antibodies. It is found in blood, lymph, intestine. Its half-life in serum is 23 days. It works in complement fixation. Whenever there is placental transfer, IgG gets placental transfer to the child from the mother. The major functions of IgG are: it enhances the phagocytosis, it neutralizes toxins and viruses, and it protects fetus and newborn. Majorly, whenever IgG antibody is there, it will be transferred from mother to the child, and thus it protects the fetus and newborn which is present in the mother's womb. Now, the another class of antibody we have is IgM. IgG has a structure of monomer, but IgM, it has a structure of pentamer. The percentage serum antibodies is 5 to 10%, and IgM are mostly found in blood, lymph, B cell surface. Whenever IgM are found on the B cell surface, they are found to be in monomer structure. Their half-life in serum is 5 days. They work with complement fixation, but IgM does not get transfer during the placental transfer. Their known functions are: they are the first antibodies which are produced during an infection. So, whenever any foreign antigen comes from our body through any viral infection through any bacterial infection or any microbial infection, the first antibodies which are produced in our body are IgM. Our immune system of the body, it gets triggered or it gets initiated whenever any foreign antigen enters our body. The foreign antigen can enter through any microbial infection, and whenever the immune system gets triggered, the first antibody which gets triggered or which gets produced is IgM to help survive us and to help us fight from that particular infection. The third class is IgA. It has a structure of dimer. The percentage serum antibody is around 10 to 15%. The location, mostly they are found in secretions of tears, saliva, intestine, milk, blood and lymph. Half-life in serum is 6 days. The complement fixation, it does not work in complement fixation, it does not get transferred during the placental transfer, and their major function is the protection of mucosal surfaces and provides immunity to infant digestive tract. So, they are formed whenever there is any digestive infection to a small child. The next class of antibodies are IgD. They are having a monomer structure. The percentage in the serum is 0.2%. Their location is B cell surface, blood and lymph. The half-life in serum is 3 days. They also do not work in complement fixation. They do not work in placental transfer, and their known functions are: in serum function is unknown, on B cell surface they initiate immune responses. IgE is the fifth class of antibody. They also have a monomer structure. They have the very least amount of percentage in serum antibodies, which is around 0.002%. Mostly IgE are found on the mast cells and basophils throughout the body. They are also found in blood. Their half-life in serum is 2 days. They do not work in complement fixation. They do not get transferred by placenta, and they are mostly formed whenever we have any allergic reactions or any possibly lysis of worms. During that time, our body produces IgE, or we can say immunoglobulin G E and antibody IgE. Now, whenever we are talking about the antibodies and antigen reaction, we talk about that these reactions are very specific. So, the reactions are specific because the antibodies structure is like that only particular type of antigen will come and react with the antibody. So, antibodies have very high specificity and very high affinity. That means, in whatever reaction they will be involved, these antibodies and antigens will be very specific reactions for a specific antigen. The antibody site is very specific and it allows the detection of analytes by a variety of immunoassay methods. So, in immunoassays, both monoclonal and polyclonal antibodies are used for the detection of antigens, each with specific strengths for applications. Immunoassays that detect antibodies in patient sera are likely to involve detection of polyclonal antibodies, which are generated by the patient's immune system. So, the antigen found could be monoclonal in nature, or the antibodies could be monoclonal or polyclonal in nature. Whenever we are talking about how the immunoassay methodology works or the immunoassay texts work, they are either non-competitive or competitive immunoassays and homogeneous and heterogeneous immunoassays. Now, what are non-competitive and competitive immunoassays? They, whenever we are having a labeled material which is And before starting with an immunoassay reaction, we require the use of a labeled material. Why there is a requirement of labeled material? Because we have to measure the reaction. The reaction is quantitative in nature. Supposedly, just to understand with the layman words, if we have 10 antibodies and we have 15 antigens, then we have to know how much antigen antibody reactions takes place by the antigen antibody combination. The remaining 5 antigen will tell us that 5 antigens are left behind, only 10 antigen and antibody reaction has taken place. So, it can tell that 10 antibodies were present because we know how much amount of antigen we have added. So, the reaction is measurable in nature, and to measure the reaction we have to make the material labeled. So, what is labeling? It is a molecule that will react as part of the assay, and a change in signal can be measured in the blood reagent solution. So, what could be a label? It could be a radioactive compound, it could be an enzyme which causes a change of color in the solution, or it could be a substance that produces light. So, the reaction could be a radioactive reaction, an enzymatic reaction, or any photoluminescence reaction. The label which can be applied, it is applied during the manufacture of the reagent, and the label could be added if we are doing specific reactions with the antigen and antibodies which are lab produced. Then antigen and antibody both can be labeled while we are manufacturing them. If the antigen or antibody, they are natural in nature, they can be labeled in a different way. So, these technologies, they utilize different formats to distinguish the bound antigen antibody complex from the free unbound label, and thus these reactions are measurable in nature. So, whenever any antibody is there and it has an antigen binding site, the antibody and antigen complex is formed. Only a specific antigen will bind to a specific antibody, which shows that the reactions are specific in nature. The binding, the making of antigen antibody complex and the remaining antigen left will also give us the measure of how much antigen or antibody is present there. As we can see in the second figure, that we have antibody and antibody reagent, and if we have added a labeled antigen to it, only the specific antigen will connect with the antibody, and how much antigen and antibody are being connected, that will give us a measurable amount that this much antigen or antibody was present in the reaction. So, these immunoassay reactions, they are competitive and non-competitive in nature. The measurement of analyte in an immunoassay is achieved by using either a competitive or a non-competitive format. Whenever we are talking about the two type of formats, a competitive which says that the antigen and antibody they fight for their specific reactions, that is a competitive format. In this, the unlabeled analyte or the antigen in the test sample is measured by its ability to compete with the labeled antigen in the immunoassay. So, what happens here is, we have a reaction. In this reaction, we have antibodies and we have certain labeled antigens and certain unlabeled antigens. So, here the competition is between those two type of antigens, and these two type of antigens both are specific to the antibody. So, which unlabeled antigen or the labeled antigen, they have a competition that which antigen will get attached to the particular type of antibody. As the unlabeled antigen blocks the ability of the labeled antigen to bind because the binding site on the antibody is already occupied by the unlabeled agent. So, in a competitive immunoassay, less label measured in the assay means more of the unlabeled or the test sample antigen is present, and the amount of antigen in the test sample is inversely related to the amount of labeled measured in the competitive format. So, just if we look at an example, if we have 8 binding sites and if we have 6 labeled and 6 unlabeled antigens, and with the competition, if all the unlabeled get attached to the antibodies, then by the remaining unlabeled antigen measure, we can tell that how much sites are being taken by the unlabeled antigen and which is the test sample which we have to analyze. So, this figure is showing us that how the amount of label measured in the competitive format and the amount of antigen in the test sample is inversely related to the amount of label measured in the competitive format. So, to conclude that in a competitive immunoassay and in a non-competitive immunoassay, depending upon the space taken by the labeled antigen and the unlabeled antigen will help us in measuring the reaction and will help us in knowing the quantity of the reaction, which is very important, where we have to know how much amount of test reagent was present whose quantity is unknown. So, these immunoassay techniques are specific, they have a very good affinity and they are also quantitative in nature, which are very good analytic technique in the cases of forensic science. Thank you.